Basal metabolic rate.
Basal metabolic rate, and the closely related resting metabolic rate, is the rate of energy expenditure by humans and other animals at rest, and is measured in kJ per hour per kg body mass. Rest is defined as existing in a neutrally temperate environment while in the post-absorptive state. In plants, different considerations apply. The release, and using, of energy in this state is sufficient only for the functioning of the vital organs, the heart, lungs, nervous system, kidneys, liver, intestine, sex organs, muscles, brain and skin.
Description.
The body's generation of heat is known as thermogenesis, and it can be measured to determine the amount of energy expended. BMR generally decreases with age and with a decrease in lean body mass, as may happen with aging. Increasing muscle mass increases BMR, although the effect is not significant enough to act as a weight loss method. Aerobic fitness level, a product of cardiovascular exercise, while previously thought to have effect on BMR, has been shown in the 1990s not to correlate with BMR when adjusted for fat-free body mass. New research has, however, come to light that suggests anaerobic exercise does increase resting energy consumption. See aerobic versus anaerobic exercise. Illness, previously consumed food and beverages, environmental temperature, and stress levels can affect one's overall energy expenditure as well as one's BMR. BMR is measured under very restrictive circumstances when a person is awake. An accurate BMR measurement requires that the person's sympathetic nervous system not be stimulated, a condition which requires complete rest. A more common and closely related measurement, used under less strict conditions, is resting metabolic rate, RMR. BMR and RMR are measured by gas analysis through either direct or indirect calorimetry, though a rough estimation can be acquired through an equation using age, sex, height, and weight. Studies of energy metabolism using both methods provide convincing evidence for the validity of the respiratory quotient, RQ, which measures the inherent composition and utilization of carbohydrates, fats and proteins as they are converted to energy substrate units that can be used by the body as energy.
Nutrition and dietary considerations.
Basal metabolism is usually by far the largest component of total caloric expenditure. However, the Harris-Benedict equations are only approximate, and variation in BMR, reflecting varying body composition, in physical activity levels, and in energy expended in thermogenesis make it difficult to estimate the dietary consumption any particular individual needs in order to maintain body weight.
Physiology.
Both basal metabolic rate and resting metabolic rate are usually expressed in terms of daily rates of energy expenditure. The early work of the scientists J. Arthur Harris and Francis G. Benedict showed that approximate values could be derived using body surface area, computed from height and weight, age, and sex, along with the oxygen and carbon dioxide measures taken from calorimetry. Studies also showed that by eliminating the sex differences that occur with the accumulation of adipose tissue by expressing metabolic rate per unit of fat-free, or lean body mass, the values between sexes for basal metabolism are essentially the same. Exercise physiology textbooks have tables to show the conversion of height and body surface area as they relate to weight and basal metabolic values. The primary organ responsible for regulating metabolism is the hypothalamus. The hypothalamus is located on the diencephalon and forms the floor and part of the lateral walls of the third ventricle of the cerebrum. The chief functions of the hypothalamus are control and integration of activities of the autonomic nervous system, ANS. The ANS regulates contraction of smooth muscle and cardiac muscle, along with secretions of many endocrine organs such as the thyroid gland, associated with many metabolic disorders. Through the ANS, the hypothalamus is the main regulator of visceral activities, such as heart rate, movement of food through the gastrointestinal tract, and contraction of the urinary bladder. The ANS regulates contraction of smooth muscle and cardiac muscle, along with secretions of many endocrine organs such as the thyroid gland, associated with many metabolic disorders. Through the ANS, the hypothalamus is the main regulator of visceral activities, such as heart rate, movement of food through the gastrointestinal tract, and contraction of the urinary bladder, production and regulation of feelings of rage and aggression, regulation of body temperature, regulation of food intake, through two centers. The feeding center or hunger center is responsible for the sensations that cause us to seek food. When sufficient food or substrates have been received and leptin is high, then the satiety center is stimulated and sends impulses that inhibit the feeding center. When insufficient food is present in the stomach and colon levels are high, receptors in the hypothalamus initiate the sense of hunger. The thirst center operates similarly when certain cells in the hypothalamus are stimulated by the rising osmotic pressure of the extracellular fluid. If thirst is satisfied, osmotic pressure decreases. The feeding center or hunger center is responsible for the sensations that cause us to seek food. When sufficient food or substrates have been received and leptin is high, then the satiety center is stimulated and sends impulses that inhibit the feeding center. When insufficient food is present in the stomach and colon levels are high, receptors in the hypothalamus initiate the sense of hunger. The thirst center operates similarly when certain cells in the hypothalamus are stimulated by the rising osmotic pressure of the extracellular fluid. If thirst is satisfied, osmotic pressure decreases. All of these functions taken together form a survival mechanism that causes us to sustain the body processes that BMR and RMR measure.
BMR estimation formulas.
Several prediction equations exist. Historically, the most notable one was the Harris-Benedict equation, which was created in 1919. The original Harris-Benedict equation. For men, for women. Where P is total heat production at complete rest, M is the weight, H is the height, and A is the age, and with a difference in BMR for men and women being mainly due to differences in body weight. For example, a 55-year-old woman weighing 130 pounds, 59 kilograms, and 5 feet 6 inches, 168 centimeters, tall would have a BMR of 1272 kilocalories per day or 53 kilocalories per hour, 61.3 watts. In 1984, the original Harris-Benedict equations were revised using new data. In comparisons with actual expenditure, the revised equations were found to be more accurate. The revised Harris-Benedict equation. For men, for women. It was the best prediction equation until 1990, when Mifflin et al. Introduced the equation. The Mifflin-Streeter equation. Where S is +5 for males and 161 for females. According to this formula, the woman in the example above has a BMR of 1204 kilocalories per day. During the last 100 years, lifestyles have changed and Frankenfield et al. Showed it to be about 5% more accurate. These formulas are based on body weight, which does not take into account the difference in metabolic activity between lean body mass and body fat. Other formulas exist which take into account lean body mass, two of which are the Katch-McArdle formula, and Cunningham formula. However, the Cunningham formula is used to predict RMR instead of BMR. The Katch-McArdle formula, resting daily energy expenditure. Where LBM is the lean body mass in kg. According to this formula, if the woman in the example has a body fat percentage of 30%, her BMR would be 1263 kilocalories per day. The Cunningham formula, RMR. Where LBM is the lean body mass in kg. Since lean body mass is metabolically active versus fat cells which need very few calories to be sustained, these formula tend to be more accurate, especially with athletes who have above average lean mass and little body fat. To calculate daily calorie needs, the BMR value is multiplied by a factor with a value between 1.2 and 1.9, depending on the person's physical activity level.
Causes of individual differences in BMR.
The basal metabolic rate varies between individuals. One study of 150 adults representative of the population in Scotland reported basal metabolic rates from as low as 1027 kilocalories per day, 4301 kj day, to as high as 2499 kilocalories day, 10,455 kilojoules day. With a mean BMR of 1500 kilocalories day, 6279 kJ day. Statistically, the researchers calculated that 62.3% of this variation was explained by differences in fat-free mass. Other factors explaining the variation included fat mass, 6.7%, age, 1.7%, and experimental error including within-subject difference, 2%. The rest of the variation, 26.7%, was unexplained. This remaining difference was not explained by sex nor by differing tissue size of highly energetic organs such as the brain. Thus there are differences in BMR even when comparing two subjects with the same lean body mass. The top 5% of people are metabolizing energy 28 to 32% faster than individuals with the lowest 5% BMR. For instance, one study reported an extreme case where two individuals with the same lean body mass of 43 kilograms had BMRs of 1075 kilocalories day, 4.5 megajoules day, and 1790 kilocalories day, 7.5 megajoules day. This difference of 715 kilocalories day, 67%, is equivalent to one of the individuals completing a 10-kilometer run every day.
Animal BMR.
Kleiber's law relates the BMR for animals of different sizes, and the observations indicate that the BMR is proportional to the 3/4 power of body mass. Warm-blooded, cold-blooded and unicellular organisms fit on different curves.
Biochemistry.
About 70% of a human's total energy expenditure is due to the basal life processes within the organs of the body, see table. About 20% of one's energy expenditure comes from physical activity and another 10% from thermogenesis, or digestion of food, postprandial thermogenesis. All of these processes require an intake of oxygen along with coenzymes to provide energy for survival, usually from macronutrients like carbohydrates, fats, and proteins, and expel carbon dioxide, due to processing by the Krebs cycle. For the BMR, most of the energy is consumed in maintaining fluid levels in tissues through osmoregulation, and only about 1/10 is consumed for mechanical work, such as digestion, heartbeat, and breathing. What enables the Krebs cycle to perform metabolic changes to fats, carbohydrates, and proteins is energy, which can be defined as the ability or capacity to do work. The breakdown of large molecules into smaller molecules, associated with release of energy, is catabolism. The building up process is termed anabolism. The breakdown of proteins into amino acids is an example of catabolism, while the formation of proteins from amino acids is an anabolic process. Exergonic reactions are energy-releasing reactions and are generally catabolic. Endergonic reactions require energy and include anabolic reactions and the contraction of muscle. Metabolism is the total of all catabolic, exergonic, anabolic, endergonic reactions. Adenosine triphosphate, ATP, is the intermediate molecule that drives the exergonic transfer of energy to switch to endergonic anabolic reactions used in muscle contraction. This is what causes muscles to work, which can require a breakdown, and also to build in the rest period, which occurs during the strengthening phase associated with muscular contraction. ATP is composed of adenine, a nitrogen-containing base, ribose, a 5-carbon sugar, collectively called adenosine, and 3 phosphate groups. ATP is a high-energy molecule because it stores large amounts of energy in the chemical bonds of the 2 terminal phosphate groups. The breaking of these chemical bonds in the Krebs cycle provides the energy needed for muscular contraction.
Glucose.
Because the ratio of hydrogen to oxygen atoms in all carbohydrates is always the same as that in water, that is, 2:1, all of the oxygen consumed by the cells is used to oxidize the carbon in the carbohydrate molecule to form carbon dioxide. Consequently, during the complete oxidation of a glucose molecule, 6 molecules of carbon dioxide and 6 molecules of water are produced and 6 molecules of oxygen are consumed. The overall equation for this reaction is: because the gas exchange in this reaction is equal, The respiratory quotient, RQ for carbohydrate is unity or 1.0.
Fats.
The chemical composition for fats differs from that of carbohydrates in that fats contain considerably fewer oxygen atoms in proportion to atoms of carbon and hydrogen. When listed on nutritional information tables, fats are generally divided into 6 categories: total fats, saturated fatty acid, polyunsaturated fatty acid, monounsaturated fatty acid, dietary cholesterol, and trans fatty acid. From a basal metabolic or resting metabolic perspective, more energy is needed to burn a saturated fatty acid than an unsaturated fatty acid. The fatty acid molecule is broken down and categorized based on the number of carbon atoms in its molecular structure. The chemical equation for metabolism of the 12 to 16 carbon atoms in a saturated fatty acid molecule shows the difference between metabolism of carbohydrates and fatty acids. Palmitic acid is a commonly studied example of the saturated fatty acid molecule. The overall equation for the substrate utilization of palmitic acid is thus the RQ for palmitic acid is 0.696.
Proteins.
Proteins are composed of carbon, hydrogen, oxygen, and nitrogen arranged in a variety of ways to form a large combination of amino acids. Unlike fat, the body has no storage deposits of protein. All of it is contained in the body as important parts of tissues, blood hormones, and enzymes. The structural components of the body that contain these amino acids are continually undergoing a process of breakdown and replacement. The respiratory quotient for protein metabolism can be demonstrated by the chemical equation for oxidation of albumin. The RQ for albumin is 63CO2/77O2 equals 0.818. The reason this is important in the process of understanding protein metabolism is that the body can blend the 3 macronutrients, and based on the mitochondrial density, a preferred ratio can be established which determines how much fuel is utilized in which packets for work accomplished by the muscles. Protein catabolism, breakdown, has been estimated to supply 10% to 15% of the total energy requirement during a 2-hour aerobic training session. This process could severely degrade the protein structures needed to maintain survival, such as contractile properties of proteins in the heart, cellular mitochondria, myoglobin storage, and metabolic enzymes within muscles. The oxidative system, aerobic, is the primary source of ATP supplied to the body at rest and during low-intensity activities, and uses primarily carbohydrates and fats as substrates. Protein is not normally metabolized significantly, except during long-term starvation and long bouts of exercise, greater than 90 minutes. At rest, approximately 70% of the ATP produced is derived from fats and 30% from carbohydrates. Following the onset of activity, as the intensity of the exercise increases, there is a shift in substrate preference from fats to carbohydrates. During high-intensity aerobic exercise, almost 100% of the energy is derived from carbohydrates, if an adequate supply is available.
Aerobic versus anaerobic exercise.
Studies published in 1992 and 1997 indicate that the level of aerobic fitness of an individual does not have any correlation with the level of resting metabolism. Both studies find that aerobic fitness levels do not improve the predictive power of fat-free mass for resting metabolic rate. Anaerobic exercise, such as weightlifting, builds additional muscle mass. Muscle contributes to the fat-free mass of an individual, and therefore effective results from anaerobic exercise will increase BMR. However, the actual effect on BMR is controversial and difficult to enumerate. Various studies suggest that the resting metabolic rate of trained muscle is around 55 kJ per kilogram, per day. Even a substantial increase in muscle mass, say 5 kilograms, would make only a minor impact on BMR. Some studies suggest that a minimum of 20 to 25 minutes of cardiovascular training per day can temporarily increase the basal metabolic rate by around 10%, owing to an increase in the metabolism of the working muscles required for recovery as well as storage of glycogen and other fuel sources used by the body like ATP and creatine.
Longevity.
In 1926, Raymond Pearl proposed that longevity varies inversely with basal metabolic rate, the rate of living hypothesis. Support for this hypothesis comes from the fact that mammals with larger body size have longer maximum lifespans and the fact that the longevity of fruit flies varies inversely with ambient temperature. Additionally, the lifespan of houseflies can be extended by preventing physical activity. This theory has been bolstered by several new studies linking lower basal metabolic rate to increased life expectancy, across the animal kingdom, including humans. However, the ratio of total daily energy expenditure to resting metabolic rate can vary between 1.6 to 8.0 between species of mammals. Animals also vary in the degree of coupling between oxidative phosphorylation and ATP production, the amount of saturated fat in mitochondrial membranes, the amount of DNA repair, and many other factors that affect maximum lifespan.
Organism longevity and basal metabolic rate.
In allometric scaling, maximum potential lifespan, MPLS, is directly related to metabolic rate, MR, where MR is the recharge rate of a biomass made up of covalent bonds. That biomass, W, is subjected to deterioration over time from thermodynamic, entropic pressure. Metabolism is essentially understood as redox coupling, and has nothing to do with thermogenesis. Metabolic efficiency, ME, is then expressed as the efficiency of this coupling, a ratio of amperes captured and used by biomass, to the amperes available for that purpose. MR is measured in watts, W is measured in grams. These factors are combined in a power law, an elaboration on Kleiber's law relating MR to W and MPLS, that appears as MR equals W, 4me1/4me. When Me is 100%, MR equals W 3/4. This is popularly known as quarter power scaling, a version of allometric scaling that is premised upon unrealistic estimates of biological efficiency. The equation reveals that as Me drops below 20%, for W less than 1 gram, MR MPLS increases so dramatically as to endow W with virtual immortality by 16%. The smaller W is to begin with, the more dramatic is the increase in MR as Me diminishes. All of the cells of an organism fit into this range, that is, less than 1 gram, and so this MR will be referred to as BMR. But the equation reveals that as Me increases over 25%, BMR approaches zero. The equation also shows that for all W greater than 1 gram, where W is the organization of all of the BMRs of the organism structure, but also includes the activity of the structure, as Me increases over 25%, MR MPLS increases rather than decreases, as it does for BMR. And MR made up of an organization of BMRs will be referred to as an FMR. As Me decreases below 25%, FMR diminishes rather than increases as it does for BMR. The antagonism between FMR and BMR is what marks the process of aging of biomass W in energetic terms. BME for the organism is the same as that for the cells, such that the success of the organism's ability to find food, and lower its ME, is key to maintaining the BMR of the cells driven, otherwise, by starvation, to approaching zero. While at the same time a lower ME diminishes the FMR-MPLS of the organism.
Medical considerations.
A person's metabolism varies with their physical condition and activity. Weight training can have a longer impact on metabolism than aerobic training, but there are no known mathematical formulas that can exactly predict the length and duration of a raised metabolism from trophic changes with anabolic neuromuscular training. A decrease in food intake can lower the metabolic rate as the body tries to conserve energy. Researcher Gary Foster, Ph.D., estimates that a very low-calorie diet of fewer than 800 calories a day would reduce the metabolic rate by more than 10%. The metabolic rate can be affected by some drugs, such as antidepressants, which may produce weight gain. Antithyroid agents, Drugs used to treat hypothyroidism, such as propylthiouracil and methimazole, bring the metabolic rate down to normal and restore euthyroidism. Some research has focused on developing anti-obesity drugs to raise the metabolic rate, such as drugs to stimulate thermogenesis and skeletal muscle. Studies of humans with 100-plus year lifespans have shown a link to decreased thyroid activity. Their resulting lowered metabolic rate is thought to attribute to their increased life expectancy. The metabolic rate may be elevated in stress, illness, and diabetes. Menopause may also affect metabolism.
Cardiovascular implications.
Heart rate is determined by the medulla oblongata and part of the pons, two organs located inferior to the hypothalamus on the brain stem. Heart rate is important for basal metabolic rate and resting metabolic rate because it drives the blood supply, stimulating the Krebs cycle. During exercise that achieves the anaerobic threshold, it is possible to deliver substrates that are desired for optimal energy utilization. The anaerobic threshold is defined as the energy utilization level of heart rate exertion that occurs without oxygen during a standardized test with a specific protocol for accuracy of measurement, such as the Bruce treadmill protocol. See metabolic equivalent. With 4 to 6 weeks of targeted training, the body systems can adapt to a higher perfusion of mitochondrial density for increased oxygen availability for the Krebs cycle, or tricarboxylic cycle, or the glycolytic cycle. This in turn leads to a lower resting heart rate, lower blood pressure, and increased resting or basal metabolic rate. By measuring heart rate, we can then derive estimations of what level of substrate utilization is actually causing biochemical metabolism in our bodies at rest or in activity. This in turn can help a person to maintain an appropriate level of consumption and utilization by studying a graphical representation of the anaerobic threshold. This can be confirmed by blood tests and gas analysis using either direct or indirect calorimetry to show the effect of substrate utilization. The measures of basal metabolic rate and resting metabolic rate are becoming essential tools for maintaining a healthy body weight.